What if your mental health treatment were personalized for your physiology? My lab studies dopamine in the hopes of achieving this vision. Now, dopamine is a molecule that you may have seen in the popular media. You've been told a lot of things about it. You've been told to optimize it. You've been told to detox from it. Maybe kind of confusing, you've been told also to boost it. And if that wasn't confusing enough, you can turn this gray dopamine into some sparkle glitter dopamine. I think all this indicates the hype, but Joe Rogan's eyes are starting to glow. So let's take a deep breath, clear this all away, and talk about the real science of this neurotransmitter that's at the heart of mental health and that Ishmael actually already introduced us to. My lab studies dopamine from the point of view of learning. Dopamine is a critical neurotransmitter for detecting unexpected or salient events in the environment and helping us adapt our behavior to changing circumstances. But it's when those learning or adaptation processes go awry that you can get a variety of psychiatric disorders. And the one that I want to talk about today is depression. Depression can be a devastating disorder for patients and families. I unfortunately know this personally. These are some pictures of my brother who passed away in 2020 after a lifelong struggle with depression and addiction. I know I'm not alone also. Depression is an extremely common disorder. After a pandemic-related rise in 2020, I can say that 1 in 4 people will suffer from depression in their lifetime. That's astounding, and we need to figure out how to solve this problem. The perspective that my lab takes on looking at this issue is to try to understand this equation. Why does stress cause depression? The fact that stress does cause depression is extremely well supported by data. And it's probably so obvious that you know it even from some of your own personal experiences. And yet, we have no current FDA-approved treatments for depression that actually target the body's biological stress response. So I think that this is a failure, but also a great opportunity. So what do I mean by the body's biological stress response? Your body makes stress hormones. They're released from your adrenal glands, which are just above your kidneys, and they're called glucocorticoids. The one that you've probably heard of is called cortisol. Cortisol can be great. It can reduce inflammation. It can coordinate that fight or flight response that allows you to respond to threats in your environment. But when cortisol is chronically or inappropriately activated, it can cause problems. So the fact that we know all this is quite useful, and yet we've failed to turn it into treatments. So the reasons for this, I think, stem from a couple of things. One is this incomplete understanding of how stress hormones actually impact the brain circuitry for reward processing, like dopamine. And the second really important point is that there's historically been this quite poor consideration of sex differences. And this is primarily due to the exclusion of female subjects from biomedical research. This is a huge problem in general, and especially for a disorder like depression that affects twice as many women as men. We cannot really solve the biology of these disorders without understanding both male and female biology. This point was really driven home to me. My lab published this paper last year. We use mice in my lab as a model system. And we studied how dysregulated stress hormones in both male and female mice could impact dopamine transmission. And we found effects in both males and females, but critically, it seemed to happen by different mechanisms. And different mechanisms imply that we need different treatments. So this study brought me to the inspiration for my one-mind proposal, where we want to look closely at how stress hormones act directly on dopamine neurons to regulate their function. And technically, this is actually a little bit of a tricky problem because of the biology of stress. Stress hormones bind to these receptors called glucocorticoid receptors, or GRs, that are all over your brain and body. They're there to coordinate a body-wide response to stress. But if I want to see what these glucocorticoid receptors do specifically to dopamine neurons, I need a really precise tool. Thankfully, that tool exists. You might have heard of this. It's called CRISPR. It won the Nobel Prize in 2020. And it's basically a little pair of genetic scissors that I can use to cut out just one gene from the genome. And I can do this precisely in terms of just targeting one gene, and I can also do it precisely in one cell type. So we can use CRISPR to delete the glucocorticoid receptors, not from all neurons or all cells in the body, but just from these blue dopamine neurons. And then we can ask how this very precise manipulation affects dopamine neuron function. Now, I've told you that dopamine is critically linked to reward processing, and so we can assay dopamine function by looking at a reward-seeking behavior in the mice. We basically just let mice ask us for sugar, like kids asking for candy. And we see how much they do it. So in control conditions, male and female mice actually do this reward-seeking behavior at about the same rate, and this is kind of our baseline. But what we see when we knock out the glucocorticoid receptor from their dopamine neurons is quite different. In males, this manipulation decreases reward-seeking, and in females, it actually increases it. So now we have this profound sex difference that we can try to understand the mechanism of. And there's a few approaches you can take, but one approach that we are planning to do as part of this one-mind proposal is that we can pull these dopamine neurons out and we can perform RNA sequencing to look at the genes that are being expressed. And we can compare genes being expressed by dopamine neurons that lack stress hormone sensitivity because they've had the receptors deleted to control dopamine neurons that still have this stress hormone sensitivity intact. We have just begun to do this work, so this is preliminary data, but the way to read this plot is that each dot is a gene. Above 0 means the gene increased in expression. Below 0 means it decreased in expression. And then above this dotted line, the dots that I've colored in are changes that we consider statistically significant, meaning that they're likely to be true. Now, again, this is preliminary, but we already have a greater than 1,000 genes in this list that are changed that we can study. But importantly, so far, we've only looked at male tissue. So a key question is how these changes will be different in the females. But even in this male-only data so far, what we have is some really promising findings. So first of all, we see changes in genes that are related to known mechanisms of antidepressant action, and I think that helps tell us that we are on the right track. And then we see a variety of other changes in pathways related to stress, cognition, circadian rhythms that we think are going to be really important and interesting to follow up on. So then the last aim of the One Mind proposal is to look more specifically at how GRs and dopamine neurons actually regulate susceptibility to stress. In normal mice, when you expose them to chronic stress, you actually get some individual variability. You get resilience and susceptible mice, but we don't know what will happen when we knock out dopamine GR receptors. And what we hypothesize is that we will change this balance of susceptibility to chronic stress, but the mechanism and the direction of action may differ in males and females, and so it may go different ways, and that will be informative in terms of targeting treatments. So the ultimate impact of this work will be a much improved understanding of the interactions between stress hormones, dopamine, and behavior that we hope will lead to new targets for antidepressants that appropriately consider sex, gender, hormone differences in the patients. And, although this may take time to translate to the clinic, really the ultimate goal of this is to bring these new treatments to patients. So with that, I would just like to thank very much the people who conducted this work, and then, of course, the funding agencies, to which I am so proud to be adding One Mind to the list. Thank you.